Hello students, today we are doing biotech principles, topic 1, terminologies and definitions. Remember, this is a crash course. This video is only for a quick revision, only for students who have already studied this topic well. If you need a detailed understanding of this topic, see another video, that is biotech principles concepts, the concepts part of this video. Okay. So biotechnology, principles and processes. Biotechnology is a discipline of modern science which uses any technology that involves biological systems or living organisms. Also, their products to make or modify new products or processes which can be useful to mankind. Right? So it is a technology. It uses biological systems or living organisms or their products. It could be enzymes, proteins, etc. to make new products, novel products or processes which can be useful to mankind. Some examples of ancient biotechnology processes. You know, milk can turn to curds, wheat flour to bread, grapes can convert to wine. These are microbe mediated processes and they are applications of biotechnology. Why? Because they use microbes. For example, milk turns to curd due to lactobacilli. Wheat flour converts to bread because of yeast. Grape converts to wine because of again a yeast, that is Saccharomyces. So these are ancient biotechnology processes which have been used even before formally biotechnology was accepted as a branch of science. Now let us see some modern examples of biotech processes. In vitro fertilization or making of a test tube baby. Here gametes are taken from the two parents and are fertilized in an in vitro situation outside the body of the mother. So the gametes are the organisms and the zygote, which is formed after fertilization, is the product. This is an example of a biotechnological process because organisms are used and the product is useful for mankind. Vaccines. Vaccines are the product which are used by making microbes. And this is an important topic of this chapter. Bacteria or yeast can be used to make vaccines which are useful to mankind. Example, the HBV or the hepatitis B virus vaccine. Correction of defective disease or genes. This is also a part of this chapter and it is done by a biotechnological process which we are going to study further. Example, treatment of the defective gene of cystic fibrosis or some other diseases. Introducing new or desired genes from the genome of one organism to another. This is a very important advancement in science which falls under the topic of rDNA technology, part of this chapter. Example, treatment of a disease like SCID. We'll do this later. Manufacturing human protein. Example, manufacturing insulin in other organisms like bacteria. So we see here that we have used organisms. It could be gametes, which are cells, organisms. It could be bacteria or yeast, and we are using these for the betterment of mankind. We are using the organisms or their products for the betterment of mankind. So these are examples of modern biotech processes. Now, the European Federation of Biotechnology has a very specific definition. What it says: the integration of natural sciences with organisms, cells, their parts and analogues for the manufacture or development of products and services for the betterment of mankind. Okay. Now for NCRT or need preparation, what do we have to focus on in this chapter? Here biotechnology refers primarily to processes which develop genetically modified organisms. their uses and applications again for the betterment of mankind. Now, what are GMOs? We're going to study in the next few slides. So, we are going to focus on what is called genetically modified organisms in a technology called rDNA technology, or the recombinant DNA technology, which will be the mainstay of this chapter. Now, there are two very important core techniques in biotechnology, especially when you want to make products on a large or a mass scale. What are they? One is, of course, genetic engineering, which we are going to study in detail. The other is maintain a sterile ambience. Let's see what these are about. Now, genetic engineering. Here in the figure given on the right side, we are seeing a bacterium. The bacterium has its DNA. A part of the DNA is being taken and inserted into another bacterium called the host cell. And what we see is that this alien DNA or the foreign DNA from one bacterium is now incorporated into the DNA of the host organism. So the genome or the DNA of the host organism has changed. It has got transformed. This is the basic principle of genetic engineering. Now, techniques to alter the chemistry or composition of the genetic material of an organism, that is its DNA or RNA, in order to change its genotype and hence its phenotypic expression. This is genetic engineering. Techniques to alter the chemistry or composition of the genetic material of an organism, that is its DNA or RNA, in order to change its genotype and its phenotypic expression. Okay, so when you change or alter the DNA of this organism, naturally its genotype changes, and if the genotype changes, it will make proteins which will also change its phenotype. We say that this organism has been genetically engineered, and later we'll call it a genetically modified organism. Now, this is done by transferring a piece of a desired gene, that is the gene of our interest, from another alien organism, a different organism, to a competent host. A competent host is one which is able to receive this foreign DNA or foreign gene, incorporate it into its genome. Now, the second very important process in large scale biotechnology, maintenance of sterile ambience. Now, ambience means the surroundings, and sterile means contamination free. That is, while you are performing an experiment, there should be no other microbes existing in the medium. Why is this important? Now, this is necessary to enable the growth of only the genetically modified host microbe. For the bulk manufacture of novel products, that is the new phenotypic proteins like antibiotics, vaccines, enzymes, etc., which we are going to make by biotechnology. Now, when you want to produce products in large quantities, this genetically modified host has to be isolated, and you want that only this host should be cultured and multiplied. So the protein produced by this host only will be made available. If this culture medium where you are culturing this host, if it is contaminated by other organisms, then these other organisms will also grow and you will get a mixture of proteins which you may not require. Hence, it's very important to maintain a sterile ambience. Contamination will allow the growth of unnecessarily unwanted organisms and the production of unwanted proteins which may be difficult to separate from the desired pure product. This will lead to a waste of time and cost and maybe not allow us to get the actual product that we desire. How can organisms be genetically modified? So let's see this process. Now, they are done by a process called genetic engineering or genetic modification or genetic manipulation. And we're going to do this by the rDNA or the recombinant DNA technology. We're going to study this in detail. Here, the genome or the DNA of the organism is either altered or manipulated and changed, which we have seen in the previous slide. In this chapter, we're going to see how it is done. Here, we are seeing a DNA of an organism and a gene which is being removed. What is happening now? This is inserted in another DNA of another organism. So this another organism, that is the host organism, will have its DNA altered. This is the basis of recombinant DNA technology. But let us understand in detail. What is a competent host? A cell which is able to receive or accept foreign or alien DNA from an external source. This is the competent host. Of course, some natural or some special artificial technique can be used. So a cell which is able to receive or accept foreign or alien DNA, that is our gene of interest, from an external source. Okay, why this is important? Because all organisms or cells may not be able to accept or receive the gene of interest. So competent host should be able to receive or accept a foreign gene into its genome. What is the desired gene or the gene of interest? The gene which is intended to be transferred to the competent host in order to alter its genome for the expression of a desired phenotype. Okay. The host organism produces certain proteins or has certain phenotypic properties, but you want it to produce some desired protein. For that, you will have to change its genes or its genome. What gene you will insert? The gene of interest, which will produce the protein of interest so that you get the desired phenotype. So that is the gene of interest or your desired gene. The gene of interest or a desired gene here is shown being cut and picked up from DNA of an organism with the desired phenotype. Right? So one organism has the capacity to produce. Now you are introducing this into your host or the competent host. The gene of interest or the desired gene is being inserted into the DNA of the host organism. So now the second or the host organism will also produce a protein which this original organism was producing. Okay. Now why do we need to do this? If the organism A was already producing this protein, why do you want the organism B or the host to produce it? There are many reasons why you don't want the original organism and instead you want another organism to produce that protein. We will see this in the later slides. The GMO, the genetically modified organism or the transformed organism, will thus possess a piece of recombinant or alien DNA in its own DNA genome. Now recombinant means which has combined with its new DNA. So this is not the original DNA. Once it has the gene, the alien gene, now this is called your recombinant DNA. Okay. The gene of interest can also be synthetically manufactured. Here we have seen that we are receiving the gene from another organism. But this gene can also be synthetically manufactured. Now in the previous slide we saw this figure. Here what are we seeing? That there is a direct transfer of the gene of one organism into the DNA of the other organism. There is no vehicle or medium required for this transfer. So this is called a direct transfer of gene from one organism to the other. But this transfer can also happen with the help of what is called vectors. Okay, so here what is shown in the second figure, we have the DNA of one organism. From this DNA we are cutting a part of it. This is a plasmid. We'll see what a plasmid is. The plasmid has a gene which is the gene of interest. This is cut by what is called molecular scissors. and this gene is inserted into another plasmid. This plasmid is now called the recombinant plasmid vector because the gene of interest is now inserted into this. So this DNA or plasmid is a recombinant plasmid vector. This can be then transferred into the host organism. When such vectors are used for transfer of the gene of interest, we say this is a transfer via a vector. Here the vector acts as a vehicle for the transfer of the gene. What is transfection? It is a process of deliberately introducing purified nucleic acid or DNA fragments or the gene of interest into eukaryotic cells from another living cell by any mechanism. There are multiple mechanisms which can be used. But what is important, when you use the word transfection, it refers to modifying eukaryotic cells. Okay, so I'll repeat. It is the process of deliberately introducing purified nucleic acid DNA fragment or the gene of interest into eukaryotic cells from another living cell by different mechanisms. What is transduction? It is a process by which foreign or alien DNA is introduced into a cell by a virus or a viral vector. So transduction refers to the mechanism of this transfer of gene, but where the vector used or the organism used is a virus or a viral vector. Example, this transfer can happen by using lambda phages. These are a type of bacteriophage, or by using retroviruses. So when viruses are used, then the process is called transduction. Transformation. The genetic alteration of an organism or a cell other than of animal cells. Remember, here we said transfection of eukaryotic cells is called transfection. Here, when we are using cells other than animal cells, that is bacteria, fungal or plant cells, these are the host cells, and genetic alteration of these cells resulting from uptake or incorporation of alien or genetic material from another bacterium by rDNA technology. This process is called transformation. So what is the result? That the host cell gets transformed. How it gets transformed? By incorporating a foreign gene into its DNA. Okay. What is this DNA now called? The DNA is called recombinant DNA. And the host cell is now said to be transformed. So we repeat, genetic alteration of an organism or a cell other than an animal cell, like cells like bacteria, fungal cells or plant cells, resulting from uptake and incorporation of alien genetic material from another bacterium by rDNA technology. This is transformation. Now vectors. What are vectors? These are natural or artificial molecules which are usually plasmids or even viruses, which may be used as vehicles to artificially transfer or carry the foreign genetic material, that is the gene of interest, into another cell, that is the host. So basically these are medium on which your gene of interest sits and is transferred to the competent host. These are vectors. So we have seen the same figure again. So here we have DNA of an organism. We are cutting a part of its DNA and taking our gene of interest. This is the gene of interest which is cut and inserted into another plasmid. This is the bacterial DNA. Once inserted, we get a recombinant plasmid vector. If you use such a vector for bringing about transformation, then the process sometimes becomes easier, because all genes may not directly enter the host organism. Sometimes they may require vectors. Now, what is a recombinant host DNA, or what are recombinants? These are DNA molecules formed artificially in a lab by artificial methods or technology, creating new DNA sequences or genes that are otherwise not found in the genome of the host. So let's understand this. These are DNA molecules formed artificially in a lab. Okay, we are talking of this kind of a process. So this DNA, this DNA is in the form of a plasmid. It did not have this gene. So it was not otherwise found, this gene was not found in this DNA. Now you're inserting this gene artificially by a lab process, by a biotechnological process. Once this gene is inserted, it forms a new DNA sequence. What is a transformant, or what is a genetically modified organism? Now we have already done this, but let's see what is the definition. A transformant, or a transformed host, or the genetically modified organism. This is a cell whose genome has been altered by our DNA technology, that is, by introducing a foreign or alien DNA into its genome. Okay, so what we've done here, first we have modified the DNA, and then we have used this DNA, the modified DNA, recombined it with a gene, so we have a recombined vector. And when this is introduced into a host, another bacterium or a eukaryotic cell, then we say that the host is transformed. And this transformed host which gets this recombinant vector is called a transformant. Okay, so cells whose genome has been altered by our DNA technology, that is by introducing a foreign or alien DNA into its genome, is called a transformant. We will see that there is an exception to this case also, where sometimes non-recombinant vectors, means those who have not been modified, may also enter the host. This is described separately in another topic, where non-recombinant means the basic vector also can enter the host, and that also will be called a transformant. However, here the gene of interest has not entered our host organism. So you can have a modified recombinant vector entering your host cells, and sometimes even unmodified plasmids or vectors can enter your host cells. This has different implications which we will see later. Till now we have seen how to modify the genome of an organism. But are there other ways in which a genome of an organism can be altered? Let's see the alternatives to the rDNA or the recombinant DNA technology. There are three, and you've studied all of them already, but let's us revise them. You can change the genome of an organism during sexual reproduction. How? We will see that. You can also change it by hybridization or even by mutation. So the three processes which can alter the genome or the DNA of an organism can be sexual reproduction, hybridization, or mutation. So what are they? Now in sexual reproduction, we have an important process called meiosis, where there can be random recombination of genes between the parental chromosomes. This happens during gametogenesis, so that the gametes that are formed, they could be recombined, they could have new DNA, and when these recombined gametes fuse or fertilize, the zygote which will be produced, or the progeny which will be produced, will have genes or DNA which are different from that of the parents. So meiosis causes random recombination, and it can alter DNA of the progeny, making it different from that of the parents. This process is very important for evolution. Hybridization. It is a process of deliberately crossing two genetically different organisms to result in progeny with different or preferred set of traits. So you have two different organisms, each one of them has got some traits which are useful. When you cross them, the progeny will have some of the desired traits from both the organisms. So that is called hybridization, and it's a very important process which is studied in other chapters in NEET. Mutation. It is the accidental, natural or artificial change in the DNA sequence by processes or mechanisms other than the above two, that is other than recombination or hybridization. Sexual reproduction and meiosis, which causes the change in the gene sequence in the progeny. This can be considered a natural process. Hybridization is done by human beings in other organisms. Human beings themselves hybridize, and they can bring about hybridization in other animals. But even other animals, either by intent or naturally, they may have different properties and they can also hybridize, producing new genetic traits. However, mutation is a process where without sexual reproduction or without hybridization, there are changes which happen in the DNA sequence. This kind of a process can also change the DNA sequence of an organism, and then it is said to be a mutation. The definition of mutation you've done in genetics. So these are some of the important mechanisms, definitions and terminologies which you need to know as a basis to the understanding of biotechnology. Okay. The remaining topics will be done in the subsequent slides.